All right, I think we see 5 people, 5 and a quarter. Someone's, there we go, 6 people.
6 students.
There we go.
That's beautiful.
And a teacher somewhere.
Is there a teacher somewhere? Yeah.
Yeah, I'm one of those.
Okay, that's Dick.
Hi, Dick.
Okay. Everybody
And
wait.
there's Chris.
All right.
Chris.
Hi.
Hi. Do you guys, before we get started, do the students want to go around and tell us who you guys are?
Sure.
Quickly?
I'm Chase. I'm a senior.
Hi.
I'm Justin. I'm a senior as well.
Okay.
I'm Trevor. I'm also a senior. I'm Jake. I'm a sophomore.
I'm Holly. I'm a sophomore. And I'm Danny, and I'm a sophomore.
Great.
All right. Okay.
Nice to see you.
And that's Chris back there.
On our lab.
Yeah. And I'm not sure who the other
Dick
two are.
Martyr.
Dick. Okay. All right.
You two have been working together, team teaching this, right?
Yes.
Awesome. All right, so you guys are ready to get started? Well, welcome, Longmont. My name is Diane Munzenmeyer. I'm the director of the National SMART Teams, and so I've been kind of working with you and your teachers on this project. We've got Dr. Chris Chunningham here, who is your cyber mentor, obviously. Dr. Tim Herman, who is the director of the Center for Biomolecular Modeling. And then, are you in the picture?
Yeah, Mark
Holder is
more than no this thing.
You want me to stay in it?
We just want to see you.
All your hand. Yeah.
Mark does all things in the CBM, but today he is going to help make this all work. Hopefully deal with our technology.
Now I can go with you.
All right. So again, thank you for helping us with this experiment. We're not exactly sure how it's going to go, so we'll just kind of play it by ear. Hopefully we'll be able to have some good conversations and answer a lot of your questions. We were thrilled with all of your questions. You came up with some really good ones, and so hopefully we'll get to all of them today. But
We can't believe they're student questions.
Yeah.
Oh.
So you're going to have to really prove to us that these are authentic student questions.
You think the teachers wrote these questions?
I don't know these questions.
No. So, but what?
We wrote them, so.
I trusted it.
We'll be the judges.
So as the conversation goes, if more questions pop up in your mind, please ask us. So, this may lead to you guys coming up with more questions, and we want to hear those as well. And we want you to know that when this is all over, you start talking about these concepts and more questions come into your head, we're going to make time when we're in Boston for the teams like you who have done the cyber mentoring, that you can actually meet with Dr. Cunningham at ASBMB and discuss these things with him in person, okay? Sound good?
Okay.
All right. Tim, you want to say a few things?
Yeah, I have a few things I want to say before we actually get to the questions. And that is, I want to say some things about the SMART Team program in general. And we really are impressed by the questions. So it sounds to me as though you guys really have dug into that paper and you've asked some really good questions. But what I want to say is life is complicated. And what we're asking you to do and what all of the SMART teams do is you take a very complicated piece of science and you're trying to simplify it into a simple explanation as to how it works. The truth of the matter is, I don't think anyone knows, even people like Chris who study this, that life is complicated. And we want you to simplify it and be able to tell a very concise, simple story. But we also want you to appreciate how incredibly complex this is and how much we don't know.
And are still learning.
And are still learning. Chris just mentioned to us that some of the things that are in that paper, maybe just one thing, is maybe not true. I mean, the authors thought it was true when they wrote it. The reviewers didn't flag it, but it may not appear to be quite as true now as it was several years ago. I don't even know what issue we're talking about.
You might talk about it. won't you?
So I guess what I want to say is our major focus, our major goal of a Smart Team program, not so much to help you learn everything there is to know about an opioid receptor, but we want you to experience a piece of science and the way that science is done, how people like Chris do the science, what questions they ask, what the data looks like, what sort of interpretations can be made from that data, what arguments are still going on amongst that academic community that's trying to understand this protein. So we want you to simplify without losing the complexity. We want you to appreciate the complexity. We don't want you to be frustrated by the complexity. Quite the opposite, we want you to be excited by the complexity. Because as undergraduates and then graduate students, that complexity is what you're going to be working on. So that's just some flowery stuff to get us started. Do you guys, is there anyone here who has have a response to that? What am I asking you?
What are you asking?
I'm not sure I heard a question.
I know. I think, yes.
I didn't. All right. All right. We're going to move to your questions.
What do you want to do? I mean, I think, Chris, why don't you decide what you want to start discussing, and we can kind of move from there.
Yeah, so as these guys both said, those are some really great questions, and I wasn't necessarily expecting such stuff that was in such detail. The stuff that we have here to talk about, I can really dork out about, so we could be here for hours. And it's all stuff that I don't do all of this type of research, but I've read a lot of papers, and I can maybe try to fill in some gaps. So I'll get through some of these questions here. All right, so I've got a couple of slides of the PowerPoint presentation that I can send you guys later. But the first question that came up had to do with what happens to the receptor, how does it engage things downstream? If we look at the receptor, compound comes in and binds, causes a conformational change about this receptor, and it causes it on the inside to look a little bit different. So it causes it to take on a different conformation. That's actually the one limitation of models like these, that these are very hard and very static. So we're actually only looking at a single conformation of the receptor when it's bound to an antagonist. Now, as you guys correctly pointed out, this receptor can bind to G proteins, and it can also bind to beta-arrestins. Now, they both, G proteins and beta-arrestins, are coming up at the bottom of the receptor. Now, G proteins really like to bind to the loops that go between these helices. So the loops between helix 3 and 4 is going to be where the G protein comes up. The next question, and it's another $1 million question, is do these things form dimers, trimers, tetramers, oligomers? And that's actually a question that not everybody knows the answer to. So there's a lot of evidence that suggests that this particular mu opioid receptor, looks like it says, okay, great, that the mu opioid receptor can bind to other mu opioid receptors in the membrane, can bind to delta opioid receptors, can bind to beta-arrestin, can bind to all kinds of things within the membrane. We have kind of indirect evidence that supports that two of these can come together and that they can actually form functional types of things. When I say indirect evidence, I mean we have assays where we artificially take receptors and put them into cells and say, if we made this system where we had tons of mu and tons of kappa, they can form dimers. One thing to keep in mind always, I'm not going to say that they exist or they don't exist, but one thing to always keep in mind when we when we review other people's research is to keep in mind, in this particular case, what does a cell look like? A cell membrane is full of all kinds of things. There are lots of mu receptors, there are lots of delta receptors, there are lots of ion channels, there are lots of many different types of receptors. And if you were to take a picture, like really get down there and take a picture at a membrane, at a surface, you will definitely find a bunch of stuff crammed in together. And so you're definitely going to see evidence of multiple proteins engaging with themselves. So the jury is kind of still out as to whether or not different receptors actually form dimers.
I think, to me, this is another example of that, the comment that life is complicated.
Mm-hmm.
You know, we tend to think of these things as individual proteins, and we're trying to understand how individual protein works.
Mm-hmm.
But in fact, if this suggestion is correct, that maybe it is the partner that they form that plays a major role in whether or not they can become active or they can signal in one way or another.
Mm-hmm.
So I actually think this is one of the more interesting things about this paper is the dimers that you talk about.
Right.
So could I ask a question now on the group, and we'll see if they can even hear us. Yeah, they'll email the answer, and the answer is going to be yes or no. The question is that the PDB file that you used, I believe, was 4DLK or DKL. And what I'm wondering is, have you seen a dimer when you open that PDB file? I only saw the monomer, and only Mark knows the magic command that will allow you to see the dimeric form in that 4DKL, DLK that we're talking about. So the question is, have you seen the dimer in JMOL? Yeah, nod, yes, no. All right, so I don't think you have. So Mark, can you tell them right now, or is this something you would have to send them in?
No, actually, it's not too hard. So a lot of different proteins form quaternary structure, or even proteins with other proteins and make like big complexes. These sometimes are available on the protein data bank in what are called biological assemblies. This is a file that's similar in the same drop-down menu as the download your PDB file. It's actually at the very bottom of it. Very bottom of that menu always has the biological assembly. And when you open one of those biological assembly files, they usually get downloaded as a zip file, so you have to unzip them. But when you open one of those in JMOL, you can right-click anywhere on the display where it's actually showing your molecule. And from that menu, there's about the second or third one down, you can select whether you want to show one model, two models, or all the models in a biological assembly. And so if you were to download this biological assembly for the opioid receptor you're working with, you'll actually see that there's two identical copies kind of interacting with each other. It does take a little bit of runaround to get the correct file from the protein data bank and go through those commands in JMOL, but a lot of different PDB files that you download that look like a single monomer will look a little different if you were to download this biological assembly.
Okay. All right. What's next?
Well, let me just finish up with the dimers. So one of the things that the paper goes into is if you look at the different transmembrane helices. So I think it's transmembrane helix 5 and 6, maybe, right? Yeah. Yep. So if you look at helix 5 and 6, is that what they were saying is that the surface of those helices would line up with itself really well. So it's going to be pretty lipophilic, and it I guess lines up as a really tight bind.
Can I suggest one different thing? I'm going to hold up a different model here because Chris has been holding a model of, I think, the delta opioid receptor. But this is actually, I hope you can see this, this is a model of the mu receptor. So this is the same protein you designed. This is the design of another SMART team, so it doesn't look anything like your design. But here I'm hanging on to the lysozyme. So I presume you all know the story of why lysozyme is fused right here between transmembrane helices 5 and 6. So this SMART team highlighted these two alpha helices, and I presume they highlighted them because they are the ones that are involved in the dimer that it forms with itself. Yeah. So this SMART team is, I presume, telling the story of how this dimerization might result in downstream signaling. Mm-hmm. And I remember reading in the paper that they said that they suspect that in the dimeric form, the G proteins are not able to bind to the dimeric form.
Right. So so the different dimers are, according to the theories, is that different dimers, and it could be mu with mu, or it could be what we call heteromers, so mu with delta, that they can actually come together. And it actually ties in really nicely with that first question I was talking about with the intracellular signals. Yeah. Is if you think about it, G proteins, and in this particular case, we'll just assume that this is not a lysozyme. We'll assume this is a G protein. Notice that these things are very large. And the G proteins themselves are heterotrimeric, and they're gigantic proteins. And so if you think about two receptors physically coming proximally right next to each other, it's really going to change what the inside of the receptor looks like. So there's a certain school of thought that says if you can create compounds that selectively hit dimeric forms of receptors, that you can get around things like the development of tolerance, or that you can get around the development of addiction. And so that's one of the things that that the paper was talking about. Yeah.
One of the things that impresses me with this whole field is, this structure only became available in 2012, and that was like just last year. Not exactly, but the point is we haven't known this for very long at all. So I wish we could fast forward 2, 4, 5 years, because if you're optimistic, then you'll say that in that amount of time we will know exactly what you just said, how we can come up with a brand new drug that can have exactly the therapeutic effect that we're looking for without the side effects that we don't want. So this is really interesting cutting-edge research, and we can only guess where it's going to.
Yeah. Yeah, and like I was saying earlier with the functional selectivity, where you can either signal through G proteins or through beta-arrestins, we are just scratching the surface of that. And so we are just learning that it's not enough to find a compound that's selective for mu. Now we have to find compounds that are selective for mu through GI, or through GO, or through GS. So tons of places to do research. Yeah.
Many questions for these kids to address when they're graduate students looking for a dissertation problem.
Yeah, there are other questions like, where do you get money to do that? Yeah, so there's also several questions about what I do. So I'm selfish. I can talk about myself all day. So it's great that we actually have mu receptors and delta receptors, because my research interest lies in both of these. So one of the biggest problems in the clinic, so when you give an opioid in the clinic, it's to treat severe pain. So severe pain from cancer, from surgery, all kinds of reasons why people are in chronic pain. The biggest problem that we see in the clinic is that a patient becomes tolerant to having the drug on board. So what tolerance means is that the same dose over a long period of time no longer produces an effect. So let's say that you break your leg on Monday and you take 10 milligrams of morphine. On Tuesday you take 10 more milligrams, and Wednesday 10 more milligrams. Kills your pain just fine. A month from now, or actually a couple weeks from now, that 10 milligrams doesn't kill your pain anymore because your body is used to having the drug on board. That's tolerance. It means that your body is doing different things to the receptors, or different things to itself that basically shut down this exogenous administration of opioids. So this is a big problem, because it means that we have to keep upping the dose of morphine that we give a patient. When we do that, all of a sudden side effects like constipation really start to come in and really muck things up. So what we're trying to do in our lab is develop opioids that we can give to patients that won't produce tolerance. So give a drug to a patient that they'll be able to take the same dose forever, and it'll be still active months later.
Can I ask a question? Do you have any idea, what are the ideas as far as the mechanism of tolerance?
There are loads, and I'm going to get into one of them.
Okay, all right.
So there are loads of reasons why a body can become tolerant to a drug. It can metabolize it down faster. It can prevent it from getting to the site of action. It can do all kinds of things. Yep. You can down-regulate the receptor so you not produce as much receptor. You can internalize the receptor. You can shut off those pathways, so those G protein pathways. Your body has ways of shutting those things down. So one mechanism that I'm really fascinated in that nobody really has a great handle on is that when you activate mu, if at the same time as giving a mu agonist like morphine, if you give a delta antagonist like a drug called Naltrindol, giving those two at the same time, for some reason, we're still not exactly sure why, produces no tolerance. So that morphine works just fine if you give it alongside of a compound that shuts off the delta receptor, like Naltrindol.
Can I ask you how you know that? Was this done in mice? Did you co-administer those two drugs to mice over a long period of time?
Yeah, it's actually other really smart people other than myself that have done this. So using several different tools, and it's always important to have a bunch of evidence. There are some people who have genetically gotten rid of the delta receptor, no tolerance to mu. If you co-administer a delta antagonist with a mu agonist, so two drugs at once, no tolerance to mu. You can give other compounds, so you can actually look in cells. You can have both of the receptors there and give a mu agonist and a delta antagonist, two different drugs at the same time, no tolerance. You can also give peptides, so other proteins that people have synthesized that simultaneously activate mu and inactivate delta. So they have structural pieces that activate this one and shut this one off, no tolerance. Peptides are a good research tool, but they don't necessarily make tremendous drugs. What we want to try to do is make a small molecule, so something just like morphine, that will be orally administrable, so you can take the drug orally just like you would take any other kind of pill that would activate mu and inactivate delta. So we're trying to make one small molecule that does both at once.
You know, I've heard you talk about this several times, and yet it really made sense this time. So that's
good to know.
I think I get it now after hearing it 4 or 5 times.
Only a couple of years. Yeah, so what we're doing in the past, what we had done is we kind of piggybacked on other researchers, taken some aspects of things that they had done for different types of projects, and we said, gosh, we can take the good bits of what you do and put it all into one. And so we've got a compound that partially activates mu and completely inactivates delta. So it's also got its place in the choir, so to speak. But what we're trying to do now that we actually know the structures or have a good handle on the 3D structures of the molecules, of the proteins, now we can rationally design compounds that would do both.
Sure. Okay. Very exciting.
So if there's any questions, please send an email my way and we can ask. Oh, here we go. Why wouldn't you keep administering both drugs? The mu agonist and delta antagonist doesn't work.
Yeah, problem solved. You've already given the solution.
Great question. Great question. And you guys are really thinking. There's actually two issues. One, and maybe it's the most rate limiting, is that Naltrindol is not approved for use in humans. We can't give it to people. We actually don't have any drugs that have been approved to give to people. So we can't give any drugs have been approved that inhibit delta that we could give to patients.
Why is that?
Lots of different reasons. Yeah, it takes it takes about $1 billion to get a drug approved in people. So that's, when I get a couple bucks, I'll work on that. But
I'm a little confused now because there's naloxone or Narcan, and in my mind, I have thought that those two approved drugs are the same as Meltrindol.
Meltrindol is specific to Delta, right?
Okay.
And Meltrindol is
All right. Yep. So that's my mistake, that they are two different drugs, or we can't use them.
Right. So naloxone or Narcan is the antidote. So if a person is overdosing actively from an opiate, you would give an injection of Narcan, and what it does is it inactivates mu, and so it kicks the agonist off and shuts this receptor off.
Okay.
So we couldn't give a mu antagonist because it would counter the effect of the mu agonist.
Yeah.
But the first reason that we can't simply do this is that the drug is not approved and it's for use in people. So,
but if it were approved,
we could absolutely do this
cocktail.
Yep.
And that would be what you'd want. Yeah.
Right. We have lots of examples of co-administered drugs. In one pill, you can have two agents. Tons of examples of that.
Okay. Right. Great question.
Yep. I guess the second answer to that is that we could give two drugs to treat one disease, but if we think about the patients that are taking these drugs chronically, like an opiate, maybe they have cancer pain or other types of pain as a part of growing older, is they're probably already taking a host of other drugs.
Yeah.
Every time you add another drug into the mix, you're adding problems with metabolism, different availabilities, and they can really be interacting with the other drugs they're taking. So the best case scenario is to create one drug that does both.
Just knock out the Delta receptor in humans.
Yeah, it's very difficult to genetically. Yeah, it's difficult to genetically knock out a person.
Yeah.
Yeah. Okay.
Good question.
No, that was a wonderful question. Now, you guys had some questions about your hooks and what we can do as part of your project. Did you have any?
So let me raise one more question before we get to their poster and hooks and so forth.
Yeah.
I saw a question. I think this was actually from another smart team, but it was something about how long does the drug remain bound? So I think this comes under the same heading of, all these things are more complicated than we like to think. I'm a very simple-minded person, so I think of a drug binding, period, done, it's there. What effect does it have? But why is that not quite all the story?
So receptor binding theory can be a very difficult and very elongated process. And we can get delta G's and free energy, and we can get all kinds of maths involved. And I don't have a dry erase for it, so I'm not going to do that. But the short answer is that a drug will bind to a receptor on the second to maybe multiple or fraction of a second time scale. And what's happening is we're just going to use delta because it's here and it's quick. What happens is, can my drug come out? Yep. Is the drug will swim along, it'll find the active site, it'll bind, and it'll come off, and then it'll bind and come off. Now, with this one, we've got little magnets here. So whenever I get really close, it comes in and it snaps. Now, you can think of the different parts of the drug molecule, which I think is another question you guys asked. You can think about the different parts of the drug molecule as kind of acting like little magnets. So if you have a hydrogen bond donor, for example, on your compound, it'll act really nicely to a hydrogen bond acceptor. So it'll bind really nicely in the active site. And whenever you get a magnet, two magnets really close to each other, you notice that you can get kind of close and then eventually they just snap because you're close enough that it gets really nice. Well, if you think about conceptually a drug coming in and going to bind to a receptor and then it wants to come off, it's going to still be physically really close to that magnet. So when a drug binds to a receptor, it binds for a moment and then it comes apart, but then it's really close, it binds again and comes apart. So the actual kinetics of a drug receptor interaction, we say things like, oh, it binds very tightly, and we think, oh, it's going to be there for like 5 minutes.
Yeah.
But that's not entirely the case. Compounds that have really high affinity have really strong magnets, so they're really going to be pulled back into the active site. Drugs that are or compounds that are of lower affinity, maybe have less of a drive to actually bind to the receptor. But how long does it stay activated has to do with that, as well as with how fast your body can push it away, how fast your body can metabolize the compound so it no longer likes to bind itself. So
you have to think of it sort of as a dynamic system in which it's binding, coming off binding, being metabolized, more of it's coming in. Maybe the dimerization or the clustering of the receptors, the concentration of the drug in that local clustered environment might be higher there than if all of those monomers were spread out over a large surface of the membrane. Yep. So once again, all the complications that we don't normally think of at first, but when you start thinking about how it really affects pain or addiction or something like that, then all of these things have to come into play.
Right. If you think about it from your body's perspective, your body is really good at trying to stay alive, and especially your brain. It likes to kick things out of there because, well, you need a brain to function. Yeah. So your body produces so many different mechanisms of clearing out outside or exogenous things. And any medication, even though we take it for a good purpose, your body doesn't know the difference. Yeah. So your body's going to be trying to actively get rid of it. Yeah. Everyone has their own style. Some people go with the less is more style, where you write like very few words and you just put up a bunch of pretty pictures. That's me. A lot of people do it that way. Some people will put as much text as humanly possible, such that a person could walk up and wouldn't even need to talk to you. I like to kind of thread the middle of that. So with me as a chemist, I do a lot of synthesis, and so my posters will have essentially structures of compounds and arrows saying this plus that gives you that, and the meat of it is really only going to be entertaining to other chemists. So I think I actually actively dissuade people like Dr. Herman from coming by and talking to me. But the thing,
have one picture of a protein there.
yeah, if you can get some pretty pictures, that'll be good. In the case of this particular project, to have pictures of proteins and three-dimensional objects, especially if they're colorful, that really draws the eye. So if you can have a picture or a diagram of the mu opioid receptor and actually pointing to different bits on your poster, I think that would be really pretty slick. And so that way you can kind of draw the eye where you want to take your willing victim as they come by at the meetings. And that's also great because you'll have your model that you can then physically show them, like, oh, you're interested in that? Bam.
That's the thing. You guys are going to have a great advantage over all of the undergraduate researchers that will be next to you at this poster session, because you'll have your physical model. You'll not only have a colorful poster, you'll have the physical model in your hand. So you guys should be prepared to be swamped by people who will want to talk to you about what is that. Yeah, why did you color it that way? Yep. So the one, if I remember the question had something to do with what is the hook? And, okay.
Yeah. For the most of our assets, we just send the opioid addiction. We have a better idea of the hook.
Okay.
That will create a better narrative for the poster. So
I think that's a fine hook, and yeah.
And today's, right now is actually opioid addiction is back into kind of the fore. A lot of times with different research topics, sometimes they're really exciting and then people forget about them. And then they're exciting again and then people forget. Opioid addiction and dependence is a huge thing right now.
Yeah.
What we're seeing, right here in Milwaukee, Wisconsin, is that we're seeing a lot of deaths from heroin. And so heroin, being an agent that acts very similar to morphine, is a big problem. And trying to figure out how to keep people from becoming dependent upon it, becoming addicted, What is it that we can do to help people who are currently addicted? These are all conversations that multiple different scientific researchers are trying to tackle. So I think dependence and addiction are perfect hooks. I think a lot of people will be taken into that.
One thing I've always been thought was an interesting part of the story is that naloxone or Narcan, and how that will very rapidly displace heroin, and therefore eliminate those downstream effectors that are causing respiratory depression and ultimately death. So it's really amazing to me that you can have two different small molecules, one heroin, which is bound, and it's basically causing people to stop breathing, and then you can administer another small molecule that is different, but not different in a way that we can easily say why it's having a different effect. But two small molecules binding to the same protein and having dramatically different effects. That's just amazing to me.
And it happens like that.
Yeah.
If you have a patient that is actively overdosing, so not breathing, not responding, a first responder, so a nurse or an emergency physician can just take a syringe full of Narcan, hit it into you, and within seconds they'll recover.
Yeah, that's magic.
Yeah.
And where did you get inspiration to study?
Yeah.
Okay. Yeah, that's actually a great question. So this is a long answer maybe. When I went to my elementary school reunion when I was graduating from high school, I told people that I was going to be a neuroscientist or something in neuroscience. And it wasn't until a couple of years ago that I reconnected with some of these people on Facebook, and they said, "Oh, well, your dream came true." Wait, what?
And you had forgotten.
And I had completely forgotten that. So I had just always been fascinated by things that act with the brain. And anything that I could do that would, I guess, get involved, like that could be involved, I thought, well, that's going to be fun. So maybe I'll try to do that. Now, when I was in high school, which is really why this conversation was odd, is when I was in high school, I really loved languages. And so I learned how to speak German, and I lived in Germany for a while. And so I went to college as a German major. So I was going to major in speaking German. Well, there's lots of people that can do that. A lot of the Germans that can do that really well. So I decided, German degree is not going to get me very far, probably. I'm sure there's lots of very good German degrees out there. But I said, not a lot of people like chemistry, and I'm kind of good at it, so that's probably going to be a good way to keep me in a job. So I declared a chemistry major, and my first year I hated it. I absolutely hated general chemistry. But then the second year,
You didn't quit and transfer to a different major.
I know, I almost did that. It was close. But
Similar experience. I majored in chemistry, and I didn't like it until I finally got the bio.
Well, for me it was organic. I really didn't care much for acids and bases and stuff like that, but the ability to be able to synthesize stuff like this and talk about stuff like this, I just thought was amazing. So when I was an undergrad, I did a lot of work. I worked in an organic chemistry lab and actually synthesized some things, and went into my Ph.D. in a pharmaceutical sciences department. So I was able to synthesize things, but then also learn about the stories and the pharmacy behind it. And so got my Ph.D., went and worked as a postdoc in a hardcore synthesis lab and in Kansas, and made my way to the Midwest, up here in Milwaukee, and here we are.
And then the follow-up question was, why the opioid receptor?
In part because that's the guy that I ran into in graduate school.
A lot of just chance encounters.
Chance encounters. So when I was in undergrad, I was thinking about being a pharmacist at one point, and I went to the University of Maryland thinking I was going to go to pharmacy school, and I happened upon this one fellow who was the only chemist in the entire school, and I said, what do you do? And he said, I work with opioids and cannabinoid, or opioids and GHB and nicotine. And I said, oh, I know about that because I took a course in psychopharmacology, so drugs that affect the brain. And so we just had a conversation. I really got into what he was doing. And so when I very first started, he said, you're going to do this, 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 and that. And then eventually, learning more about this, asking my own questions, I came up with other things to study. And so
When the agonist binds to the receptor, does the receptor change configuration and possibly make binding of the agonist more likely?
yep, that's part of that complex drug receptor energetics that that's part of the complex energetics that a drug receptor complex has. So a receptor is not, how can I say this? A receptor is plaster. Yeah, well, for one, it's not made of plaster, but it's also not always sitting in off state. And there's not just the receptor is off, and then the receptor is on. The receptor, being very dynamic, is sometimes on, sometimes off. And so it's kind of flexible, and there can be multiple on states, just as there's multiple off states. So when a drug comes around, it can actually help the receptor find the on state. So you're exactly right, is that the drug can actually help the receptor find an active form, just like the receptor being active or inactive wherever it happens to be when the drug comes in. The two kind of work together to put together a good complex. Great question.
Yeah. And we've always talked about, biology not having on switches and on switches, on and off switches, but dimmer switches. But just listening to you talk about it, it's almost, you imagine a dimmer switch, and then you imagine your grandkids sitting there playing with the dimmer switch, just turning it on, turning it off. That dynamic state you were talking about.
Well, another fascinating thing, just full of these, aren't I? Another fascinating thing is that your body can actually produce compounds that endogenously turn it on or turn it off. So what we are starting to find is if this receptor is in the membrane, you actually produce compounds that can get into the membrane and shut it off or turn it on, regardless of what's happening up here. Yeah.
So runner's high.
Yeah.
That's an endogenous.
Yep. Your endogenous mu agonist, your endorphin.
Yeah.
So there's for the different
Yeah.
there's different.
Yep. Endorphin, dynorphin, enkephalin. The dynorphin system is going to be your kappa system. So you have mu receptors, delta receptors that we've been talking about. You also have kappa receptors. Kappas are really kind of weird because they do the exact opposite of whatever mu does. So if you exercise a whole bunch and then afterwards you feel really lousy, you might actually have an overactive kappa system. So I always joke around with my wife who loves to do yoga. She just gets this huge high from it, and I say that she's really got a lot of endorphins flowing.
Yeah.
I hate doing yoga. I think I'm just like hitting a lot of kappa.
And just to make it a little bit more complicated, you go on mu and delta and kappa. There are multiple subtypes within those subtypes, or multiple isoforms, I should say, right? There's more than one mu receptor.
Some people think that.
Okay, not necessarily.
I don't necessarily agree with that.
Yeah.
There are different compounds that, again, depending on the assay, will say that will activate certain, further subtypes more. But the jury's kind of still out on that one as well. People are now more interested in these timers.
Okay, interesting. And what part of primary literature is the inaccurate part?
Well, I wouldn't say inaccurate. I would say the inaccurate part is in here when they say, where are we here? Let's see here. Well, somewhere, this paper said that if you have a drug that only signals through GI with no beta arrestin, that you would get rid of addiction. That cannot be true. Okay. Addiction is a multifaceted, complex process that even involves things like your own psychology.
So addiction is going to be in addition to your body feeling like it needs the drug, that's dependence.
Right.
Okay.
Addiction is that you feel like I need to go and take that drug to feel better and feel secure. So that right there is actually using the wrong terminology, and it came in a Nature paper.
And nobody called on that, which is shocking.
Was it sort of poorly worded?
I would say that's probably a
Yeah.
poorly worded, or we would say maybe an embellishment of what actually is going on around there. But I hedge a little bit because, you know, these are very good scientists, and I'm sure they were being true.
The crystallography paper and not a psychology. You know, it's not, they're not.
Yeah, it's the people, I mean, these people are tremendous at crystallography.
That was neural.
Yeah, and that, so the Roth group at University of North Carolina is on several of these other papers, and they do understand physiology and medicine. Yeah.
Worded it.
Mhm. Right, and I would say also, yeah, with the embellishment is things like the fact that dimers do exist and they do this. Well, we actually don't know that, so we have to say there's studies that support that. So I would say I'm not about to go out and tell you guys, nope, these guys are wrong, but I would say they're a little embellishing here or there.
I think it's important for them to know that every paper you have to read critically, right?
Mhm.
You can't just believe everything at face value.
Absolutely.
Right?
Mm-hmm. Yep.
Okay. All right. So any other questions or?
I think we're good.
I think we're about ready to call this quits.
Question? One more?
No, I think they're ready.
They're ready. Okay.
All right.
All right.
Thanks very much for tolerating us.
Yeah,
We'll
thank you.
see you in Boston.
Yep. See you.